Today we're going to talk about nerve impulses. First we'll talk about some vocabulary. So a tract is a bundle of nerve fibers in the central nervous system. Nerves are bundles of nerve fibers in the peripheral nervous system. And white matter is collections of myelinated fibers. Gray matter is collections of mostly unmyelinated fibers and cell bodies. Remember also that sensory or afferent neurons carry impulses from sense organs to the central nervous system. So that includes cutaneous sense organs that are located in your skin and proprioceptors that detect stretch or tension in muscles. And then motor or efferent neurons carry impulses from the central nervous to the viscera. So that goes to your muscles, glands, etc. So if you look at an illustration, the afferent transmission goes towards the central nervous system. So it goes through a nerve and then it goes to the spinal cord and then it can go to the brain. And efferent transmission always leaves the central nervous system and goes out towards effectors. You have several different types of nerve endings that detect various types of stimulus. So free nerve endings are the ones that are pain and temperature receptors. They're embedded in the skin and they detect pain and temperature like hot and cold. Meissner's corpuscles are touch receptors, so they allow you to detect textures and some types of light pressure. The corpuscle is this structure and it changes shape when it touches things, and that helps you to determine what texture it is that you're touching. And then you also have lamellar corpuscles. Those are deep pressure receptors, so they allow you to detect pressure of varying degrees on your skin and body. The Golgi tendon organ is a proprioceptor that allows you to detect stretch. And the muscle spindle is another proprioceptor that also detects stretch in muscles. There are several types of neurons in the nervous system that have different jobs. So an interneuron is also called an association neuron. They're found in neural pathways in the central nervous system. Their job is to connect sensory and motor neurons with each other. So in this diagram, you can see the interneuron is the one that's represented here in green in the central nervous system because it's part of the spinal cord. Another type of structural classification for a neuron is a multipolar neuron. These have many extensions from the cell body. All motor neurons and interneurons are multipolar, and it is the most common structure for a neuron. So the multipolar neuron looks like this. It's kind of that classic cell body with one long axon and then the axon terminals at the end of the nerve cell. Bipolar neurons have one axon and one dendrite. They're located in special sense organs such as in your nose and in your eyes. So they're not terribly common in adults, but they do exist. So in this diagram you can see on one side of the cell body is the dendrite, on the other side of the cell body is the axon, and the cell body resides sort of in the middle of the cell. Another structural classification for neurons are unipolar neurons. They have a short single process that leaves the cell body, and these are sensory neurons that are found in the peripheral nervous system ganglia. So those are clusters of nerve cells that are outside of the central nervous system. So this unipolar neuron has a cell body here, and then it's got this short process that attaches the cell body to the axon. The axon is a long structure, and then there are dendrites on the receiving end where it picks up messages, and then there's the axon terminals at the other end of the axon. Neurons have a couple functional properties that are important. They are irritable, meaning they're able to respond to stimuli, which is super important. And then they also have conductivity, which means they can transmit an impulse, which means they can perceive a stimulus and then they can send a message about it. When a nerve sends a message, that's called a nerve impulse. When a neuron is at rest, the plasma membrane is polarized, meaning that the charge of the solution inside of the cell is different than the outside. In this case, there are fewer positive ions inside, and there are more positive ions outside of the cell. So when you look at a neuron that's in a resting state, you can see that the inside of the neuron is slightly more negative than the outside of the neuron, which is slightly more positive. When a nerve impulse is propagated, depolarization occurs. So something or some sort of stimulus depolarizes the neuron's membrane, and then the membrane becomes permeable to sodium, and sodium channels open. A depolarized membrane allows sodium ions to flow inside of the membrane. So when the membrane depolarizes, sodium ions go into the cell from outside of the cell. This makes the inside of the cell slightly more positive than it was before. Now we have an action potential. So the movement of ions will initiate an action potential in the neuron, and it's because of that stimulus. A graded potential is when you have a localized depolarization, and that exists where the inside of the membrane is more positive and the outside is less positive, and this occurs in one section of the axon. In the diagram, you can see that one section of the axon has been depolarized, but the rest of it has not yet been depolarized. If enough sodium enters the cell, the action potential or nerve impulse can be propagated over the entire axon. So if enough sodium enters the cell, this will spread down the axon. This impulse travels more quickly when fibers have the myelin sheath. So here you can see the depolarization is spreading because it triggers more of the cell to allow sodium ions into the cell, and that causes depolarization to occur along the length of the axon. Repolarization happens in a couple steps. Potassium ions rush out of the neuron after sodium ions rush in, and that repolarizes the membrane temporarily. Repolarization involves restoring the inside of the membrane to a negative charge and the outside to a positive charge. So after the sodium has rushed into the cell, the potassium rushes out of the cell. We still need to restore the initial ionic conditions. That occurs using the sodium-potassium pump. So this pump uses ATP and it restores the original configuration. And so it ejects sodium ions from the cell, and 2 potassium ions are pumped in every time 3 sodium ions are pumped out. So this restores the initial polarization conditions. So here you can see a sodium-potassium pump is a protein that's embedded in the membrane, and it pumps sodium out of the cell 3 at a time and potassium into the cell 2 at a time. Once the nerve impulse reaches a synapse, it can send its message onto a new cell. So when the action potential reaches the axon terminal, the electrical charge opens calcium channels at the terminals. So in this diagram, you can see the action potential has arrived at the cell, and it's going to allow these vesicles, or stimulate these vesicles, to fuse to the cell membrane. When those vesicles fuse to the cell membrane, that's caused by calcium, and it allows the neurotransmitter that is in those vesicles to eventually be released. So here you can see a close-up of the vesicle fusing to the cell membrane here, and then here you can see the neurotransmitter being released into the space between the transmitting neuron and the receiving neuron. That calcium is important because it causes pore-like openings to form, and it releases the neurotransmitter into that interstitial space. So again, the neurotransmitter is released because the plasma membrane has fused with the vesicle carrying the neurotransmitter. Then the neurotransmitter molecules can diffuse across the synapse, which is the junction between the 2 nerve cells, and that binds to receptors on the membrane of the next neuron. The receiving neuron has ion channels that have receptors that the neurotransmitter can bind to, and when the neurotransmitter binds to those channels, it opens them and makes them permeable to ions. If enough neurotransmitter is released, graded potential will be generated, which means that the nerve cell can depolarize, so eventually an action potential will occur in the neuron beyond the synapse. So it will occur in the new neuron or the receiving neuron. Again, that neurotransmitter binds to the ion channel. When the ion channel opens, now it becomes permeable to sodium, which, remember, is the ion that has to travel across the membrane in order to propagate a new action potential. This process happens very quickly. The electrical changes that are promoted by the neurotransmitter binding are super short, and the neurotransmitter is quickly removed from the synapse, and it can be broken down by an enzyme. So here you can see the neurotransmitter has been broken down by an enzyme. It's released by the protein, and then it's available to start a new action potential.